synthesis, and what we decided to use with the food was like an Italian food kind of theme. So as you see, we use pepperoni,
Pizza boxes.
and we put it in pizza boxes, and we use pepperoni, cheese, shrimp, and this is made out of pizza dough. So protein synthesis.
Well, as you can see, just to explain a few things in our model, these breadsticks, these represent DNA, and the ones with the red food coloring, these are the RNA. And then we have a few different bases. We have sausage, which is adenine, shrimp, which is thymine, cheese, which is cytosine, ham is guanine, and then the different one is the olive, and this is on the RNA, and this is uracil.
So before we start talking about protein synthesis, I'm going to talk a little bit about RNA and the structure and function. There are a few differences between RNA and DNA. First off, it has, instead of thymine, as in a DNA strand, there's uracil in the RNA. Also, RNA strands usually occur only in one strand rather than two, as in DNA. And also, they're a lot shorter. So
first, what happens is this piece of pepperoni, it represents the RNA polymerase, and what happens is it comes to a strand of DNA, and then it meets the promoter, which is basically a nucleotide sequence that tells where to start, and then it begins to split the DNA apart, and then it will bring in complementary nitrogenous nucleotides, one nucleotide at a time, and bond them to the complementary nitrogenous bases. And you can see that's what's happening here with the RNA being added to the DNA.
So once the polymerase exits, basically what it does is it separates this so it's its own strand, and it puts the DNA back together. So you have two separate strands, one RNA and one DNA.
And it stops at the termination signal, which basically tells the polymerase to release the RNA.
Then what happens is this RNA strand, it goes through the nuclear membrane, nuclear envelope, and it comes over here, where this is supposed to represent the nucleus. It's outside of the nucleus, and it looks for ribosomes. So what happens is it binds to a ribosome, the small part first, and then it binds to a bigger part.
This is the ribosome. It's a pizza bagel. And then what happens is, as you can see, this is the start codon, which is AUG, and a tRNA, which is represented by the two mozzarella sticks in a T shape. It will come and it'll bring the complementary nitrogenous bases to the AUG, and it will also be carrying as you can see from the different colored dough balls, these are the amino acids, and this one is methionine, and it will bring it in, and then
another tRNA comes with the complementary bases for the next sequence, which is called a codon. And the parts on the tRNA is called the anticodon. So basically, the anticodons and codons match up. So then, what happens is, as one comes in, this one leaves, leaving the only the amino acid binded to the other amino acid by a peptide bond, as represented with the toothpick.
So then, basically, this process happens over and over. As you can see, the amino acids keep getting longer and longer. And then finally, it would reach a stop codon. And in our particular example, the stop codon is UAA, but it can also be
UGA or UAG.
And then basically, the tRNA goes away, and then you are left with a string of amino acids. And this would basically fold into a three-dimensional protein shape that could be used in various parts of the cell. So that's basically it.